So our last two slides of chapter 3 as we talk about organelles. And mostly organelles are membrane bound, but ribosomes weren't membrane bound, but you realize we have to have ribosomes. And if I were to ask someone to tell me what is the main job of a ribosome, what would you tell me? The main job of a ribosome is to do what for the cell? Make proteins. That's the job, make proteins. Now we've got a lot of other membrane-bound part of our endomembrane system, the nucleus, the endoplasmic reticulum, the Golgi, all those vesicles. But we also have these components, and these are proteins. Just like your body must have a skeletal system to maintain its shape, to allow us to do work, so your cells must have an internal framework. And that internal framework is made up of these three proteins: microtubules, microfilaments, and intermediate filaments. Now, you can think of these as cables, interconnected cables, surrounding the cell, going through the center of the cell. Some of these cables are almost like rail systems that organelles can be transported on with the use of other proteins. But what I want you to know with these three components, and that's kind of a number. We stick with kind of three, right? That's pretty simple, three. We have three chemical bonds, right? What was our weakest chemical bond? Hydrogen bonds. What was our strongest hydrogen bond? Covalent. And the one in the middle? Ionic. So similarly, when we look at our cytoskeletal proteins, microtubules, microfilaments, and intermediate filaments, these are largely defined not just by the proteins they're composed of, but by the size of the fiber, the diameter. And microtubules are the largest diameter cytoskeletal component. Tubes are pretty big, and you can think of it like that. Microfilaments have the smallest diameter. So just like our chemical bonds, we had a strong, weak, and one in the middle. If microtubules are the biggest and microfilaments are the smallest, where are you going to put the next one? In the middle. What's another good word for middle? Intermediate. You can have middle schools or intermediate schools. Don't you love it when they name something that it makes sense? Well, intermediate filaments are called that not because they're in the middle of some other filaments, just because they're halfway the diameter between microtubules and microfilaments. That's organelles, and I don't know why it keeps cutting that off there. But as we get into chapter 17 and we begin to look at cell division, we're going to see which of these components are being used to help our cells divide and help us in growth and healing. And in fact, we're going to see that the microtubules and the microfilaments are going to be important for those processes. So here's just an illustration showing the diameter difference. Here we have a microtubule, and you see how you have these spiraling monomers. These are called tubulin monomers, not that we're going to be responsible for that, but they make this spiraling fiber that has an outer diameter of about 25 nanometers. Here our microfilament has a 7 nanometer outer diameter, so it's very small. And then what do we have in the middle at about 12? Intermediate filaments. Different arrangements composed of different proteins, all functioning to help maintain the shape of a cell, help with cell motility, and then helping with cell division. Those are the big jobs of our cytoskeletal proteins. Now, it's kind of good that we finished with that, because as we move into chapter 17, into cell division, we're going to use those. We're going to have to replicate all of our organelles. We're going to have to produce more plasma membrane. But to get this one parent cell to divide into two daughter cells, we're going to have to use those cytoskeletal components. So during this process, we absolutely must have 2 copies of all of our genetic material. 1 copy is going to go to one daughter cell. The other copy is going to go to the other daughter cell. Those have to be perfect copies. Those must be complete copies. If not, the resulting cell that has too much or too little isn't going to function. Now, you know mutations, right? You've heard about mutations? And mutations in the wrong genes can lead to what horrible disease? Cancer, right? But did you know if you have too much genetic material, that can cause a lot of trouble too? You ever heard of Down syndrome? Down syndrome is also called trisomy 21, which means instead of 2 copies of 21, you have 3. What's wrong with that 3rd copy? Not a thing. It is a perfectly normal chromosome 21. There's just too much of it. And look at the degradation of your quality of life just by having too much of a good thing. See, that's why I say too much of anything is not good. Even the good stuff, it's not good. So as we look at this cell division and copying of our cells, we have 2 ways in which we can do this. Mitosis, and this, even though we've got an E in there, is called meiosis. Now, literally speaking, these are going to be means of dividing the nuclei. When we get to the point of actually dividing the cell and the cytoplasm and the plasma membrane, we call that cytokinesis. But for all intents and purposes, a lot of people equate mitosis and meiosis with 2 cell divisions. Mitosis. This is the division of your body cells, or what we call somatic cells. Soma in Latin means body. So the vast majority of cells in your body, when they replicate, they are going to undergo mitosis. There is only one population of cells in your body that undergoes meiosis. Whoops. And that's going to be the cells that give rise to either the egg or the sperm. Mitosis is cloning, exact copies. Meiosis is going to result in daughter cells that have half as much DNA. How can you get away with having half as much DNA? Because you're going to get the other half from the other partner. Okay? You can't have an egg that has a full complement of DNA and then a sperm come along. Now you got 3 times as much. That's not compatible with life. So before we get too far into mitosis, and we're going to start with mitosis first, we're going to need to learn some terminologies. And the first thing we want to understand is, what is a chromosome? And our definition is simply going to be one continuous linear nucleic acid molecule. And it is compacted down into a picture that we typically see looking like this. And what helps compact it all down are a lot of proteins that are associated with our DNA. So a chromosome is one molecule of DNA and all its associated proteins. Now, have you ever seen a picture that looked like that? What do most people call this? That's what most people call a chromosome, right? That X-shaped thing. This, in fact, are 2 chromosomes that are still attached to one another in the middle, and that middle place is called the centromere. Now, each one of these individually, these chromosomes, we could refer to them as a chromatid. But when they're attached together, they're referred to as sister chromatids. We don't refer to these as chromosomes until they separate and moving apart toward the end of mitosis. Does everyone understand the difference? A chromosome is one molecule. A chromatid is one chromosome attached to its identical copy chromosome, And since they're attached, we refer to them each as chromatids, and collectively together, that whole package of the two copies are called sister chromatids. It's important to understand this because we can't reproduce the cell, we can't have the cell divide if we don't have two copies. Oh, and there's our definition of that. Any questions? Again, as we move into this, there's going to be a lot of choreography, a lot of moving around. If you get lost, don't let the bus leave you by. So I'll get caught up now.
So if they're attached, they're chromatids, but if they're not attached, it's chromosomes.
Exactly. If they're attached, they're chromatids. Only when you have a single copy separated does it become a chromosome. Okay? Now we're starting simple and we're going to get complicated, right? Because I'm going to change that rule on you when we get down the road. But for right now, single chromosome, double chromatids. All right? Yeah.
And a chromosome is just a single strand of DNA and its proteins?
Correct. One single strand. You have multiple strands of DNA in your nucleus, and we're going to get to those numbers in a bit. Okay? But it's just one. So what we're going to do, and don't worry so much about these names right now, but when you first produce a daughter cell, after mitosis, that daughter cell is going to have a number of these individual chromosomes. And when that cell is instructed to make copies, it's going to make a copy. They're going to still be attached together. And when they're attached together, what are they? Chromatids. I don't even like it when they put replicated chromosome. I wish they would put that in parentheses, but it's a sister chromatid. And then the process of mitosis is going to be used to pull those chromatids apart into individual chromosomes. One's going to go to one daughter cell, the other's going to go to the other daughter cell. All right, so that's why we're spending a little bit of time trying to understand the players that we're going to be moving around in the process of mitosis. Now we said that a chromosome is the DNA molecule with the proteins, right? Well, if we look at our illustration, we can see our double helical DNA molecule. Well, to package that DNA down into its most compacted form, which are those chromosomes that we can see during mitosis, the beginning of the packaging starts as that DNA molecule wraps itself, or think of it like a spool. You know what I'm talking about? Have you ever seen a spool of thread? The DNA spools itself around a collection of proteins, and these proteins are referred to as histones. And when you look at this sort of conglomeration of the thread of DNA around the proteins, it's going to look like kind of pearls on a necklace under a microscope. We call that whole collection of the DNA and the protein right there, that is referred to as a nucleosome. And we're going to have hundreds and thousands of these on each chromosome, helping it compact down. So this is sort of a perspective illustration. We see our individual histone proteins. We see our double helical DNA. We begin to twist the DNA, and now it spools around our histones. What do we call that bead? The whole thing? A nucleosome. And you can see they're about every 200 base pairs, there's going to be another nucleosome. And then those start wrapping around each other, spiraling around, forming a fiber. That fiber is going to wrap around, forming a thicker fiber. Those fibers are going to loop around. Do you see how we're compacting it all down? When the cell's active and doing its thing and surviving, the DNA has to be dispersed so that you can copy the DNA and make proteins. But for cell division to occur, we've got to compact it down to make sure that this half is going one way and this half is going the other. Now, A chromosome is how many DNA molecules? One linear DNA molecule intact. And when we look at species, species are defined by how many chromosomes, how many different chromosomes compose their genome. How many different chromosomes do they have inside of their nucleus? And for human cells, how many chromosomes do we have? 46. Now that 46 is the total number of chromosomes. How many of those do you get from dad? 23. How many do you get from mom? 23. So it's okay for you to think of that 46, but I would much rather you think about the 23 number. Because you're going to get chromosome number 1, chromosome number 2, chromosome number 3. You're going to get 2 copies of each one of those. Those chromosomes are going to have the same genes on them. And when I say the same genes, chromosome number 1 may have a hair color gene. It may have an eye color gene. But the chromosome 1 hair color gene you get from mom, is it necessarily going to be the chromosome 1 hair color gene you got from dad? Those differences in the genes are called alleles. We're going to get to those in a minute. So do you understand? The 23 chromosomes are the same genes, but maybe different flavors of the genes that you get from mom and from dad. But you got 2 copies of each. Now the fact that we have 2 copies of each of those 23, you will often see that written as 2N, N being the number of chromosomes, the number of different chromosomes you have in your genome. And when we have these 2 copies, that's referred to as a condition called diploid. Now, in the case of trisomy 21 for Down syndrome, in reference to chromosome 21, that would be triploid, TRI, for 3. Now, do you realize most embryos that have 3 copies of chromosome 21 spontaneously abort? I think it's upward of 50 to 60% of embryos that have Down syndrome spontaneously abort. When you see so many living people with Down syndrome, maybe that gives you an idea of how frequently this occurs. And hopefully we'll have enough time into the semester to talk about risk factors of Down syndrome, but are you familiar with the main risk factor of Down syndrome? The one thing that can cause a greater incidence of Down syndrome in your baby? Being older when you get pregnant. Maternal age. As you get older, the incidence of Down syndrome goes up. Guys, you're not off the hook either because data's coming out that paternal age plays a role in Down syndrome. Not as significantly as maternal age. So, have the babies while you're young, right? No, I'm kidding. So once again, we've got 1 copy, and we're going to say this is chromosome 1. All right, 1 copy from dad. They painted it blue. I wish that had been pink, but this is chromosome 1 from mom. Right? So this individual single thing that I have here, what is that called? A chromosome. During the process of making our copies, as we get ready for cell division, see, we make a copy and it's still attached to each other. What is it called now? Yeah, it's not a chromosome anymore, it's a chromatid, and together they're sister chromatids. It's like they're holding hands, okay? Now, here's where I told you I was going to make things complicated. Because I said a chromosome is one thing. Okay. Well, as we move on and we talk about the 2 sets of each, now the terminology gets a little muddy because when you look at chromosome 1 from dad and chromosome 1 from mom, those 2 things are referred to as homologous chromosomes. Do you understand the homologous thing? What does homo mean? Same. So we have the same genes on each one of these chromosomes, even if the genes are slightly different. for hair color, eye color, blood type, that sort of thing. But as long as they're the same genes, chromosome 1, we call them homologous chromosomes. Now, this is what I hate. I hate. And if you want to change it, you can. If I ever write a textbook on this kind of stuff, I want to change the terminology. Because once this stuff replicates, these aren't chromosomes anymore. But look what they still call them, even when they're chromatids. They still refer to them as homologous chromosomes. I wish they would refer to them as homologous chromatids. But that's just the way they've decided to do it. So just make a note that this circumstance is a little bit tricky to use that term chromosome. Okay? This is, sometimes when you look at science and pictures of scientific stuff, there's a beauty to it. And I really like how they have taken a picture of maybe not every human chromosome. I don't think we have 23 pairs there. But when you look at what your chromosomes may look like if they were colorized as the cells are undergoing division, we can see this pair of sister chromatids, right? This is the same color, so they're saying, okay, these are our homologous chromosomes. Can I do that? Our homologous chromosomes, because I don't want to call them chromosomes, but you see here, they're calling homologous chromosomes, and these are homologous chromosomes. Okay? But I hate it when they tell you, oh, this is a rule, call it this, and then they break the rule. It drives me crazy. So 23 pairs of chromosomes is what equals our number 46. Now, of those 23 pairs, we have 22 that are dealing with the functions of your body. And we call those autosomes. But the 23rd pair we refer to as the sex chromosome. Because you're either going to have an XX or you're going to have an XY. I had 3 daughters, and my wife took great delight every time I said I wanted a boy. She took great delight in saying, oh, well, that's yours. That's your job. Right? Because if you have a son, where does that Y chromosome, where does that have to come from? Dad. What is the only chromosome you can get from mom? The X. Yep. So if there's a boy, that's going to be the Y chromosome. And did you know that people already, the technology exists for people to go to a fertility clinic and determine the gender of their child before they have a baby? Did you know that? You know how easy it is? What would be the difference between a sperm with an X chromosome and a sperm with a Y chromosome? Just a basic, logical, not a big fancy scientific difference. What would be the logical difference between a sperm with an X chromosome and a sperm with a Y chromosome? Yeah, that's going to be the result, but just think about the physical chromosome. The X chromosome is bigger. So the sperm with the X chromosome in it's going to be heavier. So all you have to do is have Dad go in the little room with his cup, you know what I'm talking about, get the sample, put it in a centrifuge with a media that has a certain buoyancy, and spin it subjected to a centrifugal force. The heavier X sperm go to the bottom. The lighter Y sperm float to the top. You either take the top fraction, artificial insemination, bottom fraction, artificial insemination, 100% chance of either a boy or a girl. All right, so now, let's get into our cell cycle. Now, with cell cycle, I want you to understand, mitosis is a part of the cell cycle. So if you've ever studied animals, you know the life cycle of animals? This is going to kind of be like a life cycle. And so, if you got to pick a place to start, we're going to start right after mitosis finishes and we have a brand new cell. This brand new cell goes through this major part of the cell cycle, and if you follow this light part around the outside, this major part of the cell cycle is called interphase. When you ever hear that prefix inter, you know what that means? What is an interstate, an interstate highway? An interstate goes between states. So interphase is the part between what? Mitotic events. So the part of the cell cycle that's between one mitotic event and the next mitotic event, somebody decided to name that interphase. And you can see mitosis is a very short period of time. The bulk of the time the cell is going to be in this interphase. And with interphase, we divide it up into 3 sub-stages. G1, S, and G2. And if you look at the little definitions of these, you can probably figure out what the G's mean. What does the G stand for? Growth. Cell's just getting bigger. You had one cell that just broke in half, so yeah, we need to grow and get bigger, make more stuff. We're going to continue to grow over here. Not only that, we're trying to get a little bit bigger because we're about to cut ourselves in half, so we need to grow. But in between our growth periods, we have this S phase, and that's not Superman. S is for synthesis, replication. And what is being replicated in S phase? DNA. So here in G1, our DNA are packaged into what can we call those molecules in G1? Just finished mitosis. We've got these individual DNA molecules, chromosomes. But once they're duplicated, they're going to stay stuck together. So what will we call them in S? Sister chromatids. What are they going to be in G2? Still sister chromatids, because we don't pull them apart until we get toward the end of mitosis. So interphase involving 2 growth periods with this middle part, very, very important part, where we actually duplicate and replicate our DNA. Now again, you can see up here how they got mitosis. Again, mitosis, strictly speaking, is just this part, the division of the nucleus, that is followed closely by cytokinesis, cyto meaning cell, kinesis and kineme meaning to move, or in this case to break apart. That's going to be the division of the cytoplasm and the cell membrane. So there's sort of some overlap there, because by the time we finish, we've divided the nucleus and we've divided the cell, the cytoplasm. So interphase, as we saw in our illustration. I mean, I don't know, what do you think? 80+% of the cell cycle is interphase, longest portion. And the major thing we're doing, we're making the cell bigger and replicating our DNA. And again, we can see the stages in order that we've written out here. G1, growth of the cell, synthesis, replication of our DNA. If you want to put out here in parentheses, sister chromatids, because that's the first place we see our sister chromatids. And the cell prepares to divide, we still have sister chromatids here. I don't know if you want to put that there or not. But we're not going to form our chromosomes again until toward the end of mitosis. All right, so this is a nice illustration of how we have our single chromosome. We begin to replicate our DNA, and so by the time we get to the end of the S phase, we absolutely have our sister chromatids. You see how they're all loose here? They're all spread out. Toward the end of interphase and the beginning of mitosis, those histones, nucleosomes, fibers are going to form, and all these linear molecules are going to compact down, and we're going to see our sister chromatids that are going to be pulled apart into our individual chromosomes, and we do it again and again and again as long as cells are dividing. So, interphase had our 3 stages, G1, S, and G2. Now when we get to mitosis, this nuclear division, there are 4 major stages. A lot of science textbooks now throw in 5. I ignore this 5th one. I just don't like it because it's kind of in between 2 of them. I like to stick with 4. It's a smaller number anyway. Prophase, metaphase, anaphase, and telophase. Those are the 4 stages of mitosis in the order in which they occur. Pro usually means before. So you can know just from the name, that's first. Meta kind of means toward the middle. So that's where instead of looking at those prefixes and thinking about what the Latin root is, it can help you out. This you kind of have to commit to memory. But one of the ways I like to commit these to memory is just simply by looking at the first letter, PMAT. Remember PMAT, that is the stages and order of mitosis. Now, I mentioned as we finished chapter 3 in cytoskeleton that we were going to use some of those fibers in cell division and mitosis. And so for mitosis, what we're going to use first are going to be a set of microtubules. And among other things, these microtubules, as they assemble and disassemble, almost like a telescoping pole. Have you ever seen those things before? These microtubules are going to be responsible for attaching to our sister chromatids, moving them around, and then eventually pulling them apart and moving them to opposite ends of the cell.
And those microtubules respond out of the centrioles, right?
Correct. We're going to get to that picture when we get to our specific phases. Those are the areas that organize these individual monomers, tubulin monomers, and organize them into our microtubule. So these microtubules that are going to go from North Pole, South Pole, East Pole, West Pole, however direction you want to do it, they're going to reach toward the middle. To me, it almost looks like a bird cage. And those microtubules that are going from pole to the center and attaching to those sister chromatids, collectively we call that the spindle apparatus. And so here in our illustration, you see these lines that are coming from the poles, and these are in fact our microtubule organizers called centrioles. We'll talk more about those in a minute. But they're extending from those all across the cell, but they're more importantly attaching to the centromeres of our sister chromatids. And it's these microtubules that push and pull these until they get right along the middle of the cell. And then these centrioles, these microtubule organizing centers, start reeling them in like you've gone fishing. And as you begin to shorten these microtubules, it pulls the chromatids apart into individual chromosomes. And as we get to the end, when we have them fully, our chromosomes at the opposite poles, we start to reform a nuclear membrane. And while we're finishing mitosis, cytokinesis, that division of the cytoplasm in the cell, begins. So there's a little bit of overlap before we finish each one of those. That's the overview. We good with the overview? All right. So PMAT, prophase, metaphase, anaphase, telophase. We're going to look at some of the features and characteristics. I like this. This is from my amoeba sisters. And honestly, if you get on YouTube and you type in amoeba sisters, mitosis, they have great, fun videos that go through the mitotic steps, the mitotic events. And like I said, they're fairly detailed and complete, even though they're using cartoons. All right? So the first phase of mitosis, P, prophase. What are some of the important events that happen? Well, I'm going to start at the bottom, actually, because the nuclear membrane dissolves. It disappears. Why do you think it'd be important for the membrane to disappear? You got to get to the chromosomes, right? The membrane would be in the way, so it's got to get out of the way. The nucleolus, that dark spot in the middle, that dissolves. Those materials spread out. So it appears to go away. But what's happening more and more as we go from interphase to prophase and later in prophase, our sister chromatids condense. They twist tighter and tighter and tighter into those fibers and those loops and compact themselves down until we can visualize them as these X-shaped sister chromatids that are still attached at the centromere, the middle part of our sister chromatid package. Now we're condensing these down. We've got our nucleus out of the way, which we have to do, because out in the cytoplasm where we have our cytoskeletal elements, microtubules, we begin to see the formation of centrioles. These centrioles are going to look like two dashes, one up and one kind of going the other direction, almost perpendicular to each other. These are what are referred to as microtubule organizing centers. They're going to assemble the microtubule monomers into these polymeric fibers that are going to form the spindle fibers and the spindle apparatus. Now, I love to show pictures because I'm a picture person. I'm not going to ask you to identify or label any pictures on the test. You do that kind of stuff in lab, all right? This is just going to be reading the multiple choice questions and picking the best answer. But in prophase, we can see that our nuclear membrane is largely gone. We can begin to see our chromosomes condensing. And at that point, we know we are looking at a cell that is in prophase. Now, the new science textbooks love to throw in an extra stage that's called pro-metaphase. It's somewhere between prophase and metaphase. I don't like it. If it's not metaphase, it's prophase. And until our sister chromatids get lined up right along the middle of the cell, we call that the equator. If these homologous chromosomes are not lined up on the equator, it's still prophase. They're moving around, they're getting close, but until they line up, it's not metaphase. Because the M in metaphase to me stands for middle. That's where the sister chromatids are, lined up in the middle. And what's pushing and pulling the sister chromatids around to get them to line up in the middle? What's doing that? The microtubules, but that larger structure is called the spindle apparatus. That's what's causing them to line up in the middle. And here you can see in our illustration, we have our centrioles out here. We have some of these other fibers that look like stars kind of spreading out. And some of these fibers are actually going to push the cell apart, make the cell more elongated. But we can see that dark line right down the middle. Those are our sister chromatids lined up right along the equator. Now, here's kind of a trick question. Think before you call out. We've got these lined up perfectly along the equator. How many sister chromatids are lined up side by side by side along the equator? How many? I heard somebody say it. 46. And they're lined up along the equator in this direction. You see they're almost perpendicular to the direction of our spindle fibers. Because this fiber is going to be pulling in this way, this fiber is going to be pulling in this way. They have to be pulled apart into chromosomes. This is going to be where we're going to form the nucleus of one daughter cell. This is where we form the nucleus of the next. How many chromosomes does each daughter cell have to have? 46. So you've got to line all 46 up along the middle. Otherwise, there's going to be an imbalance. So if they're in the middle, it's M for metaphase. And then our cells pause for just a second. We're not going to get into a lot of detail, but in the cell cycle, there are a lot of places where there's a pause, and the cells check themselves out. Have you ever watched those launches of the space shuttle and those rockets and everything? And every now and then they'll have a hold on the countdown, and they'll say it's a built-in hold. Those holds are so they can check and make absolutely sure everything is just right. Well, in this case, the pause that happens, the cell is making sure that all 46 are lined up exactly where they need to be before they try to pull them apart. That's the pause. That's the check. Because if one of these wasn't attached to a spindle apparatus, to a microtubule, and if it didn't get separated, now both copies are going to move in the same direction. That's how you get trisomy 21. When you don't have a disconnection of those sister chromatids. All right? So that's why that pause, it seems like, okay, well, what is it doing? No, you're just making absolutely sure before we get to the launch pad and blast off. And that's anaphase. As soon as the sister chromatids separate, even just a little bit separated, start moving in opposite poles, we now refer to them as chromosomes, and a lot of people have adopted this term daughter chromosomes. I kind of like that, sisters, daughters. And why did they separate, and why are they moving in opposite directions? The centrioles, the microtubule organizing centers, are reeling in the microtubules to pull those daughter chromosomes in opposite directions. Now, like I said, here in anaphase, we're also going to have elongation of the cell. It's going to go from a round cell to an oval-shaped cell. And toward the end of anaphase, this is the point in time in which cytokinesis begins, the division of the cytoplasm. Now, once the chromosomes reach the poles, at that point, telophase. When they've reached their destination, we're at the end, telophase. The chromosomes begin to relax, they uncoil. We're going to see our nuclear membrane begin to reform. The nucleolus begins to develop. The spindle fibers, we're going back to normal. Everything gets thrown in reverse from a cell standpoint because the cell is getting ready to split off from each other in the process called cytokinesis. Do you see how we're pinching together here in the middle? And here in this illustration, we can see the cells beginning to pinch off, our DNA's uncoiling. We still have a few spindle fibers that we can see in the middle, but we're just about ready to form two completely new, brand new cells that are identical to each other. Mitosis is cloning. You're making exact copies. Two exact copies from one. And so there it is all together. Interphase, cell just happy doing its thing. Prophase, we get the condensing. Nucleus breaks down, there we form our centrioles and our spindle fibers begin to form. Once we get to the midline middle, what do we have? What phase? Meta, M for middle. What gets separated in anaphase? Sister chromatids are separated into what? Chromosomes are daughter chromosomes. And in telophase, they've arrived at the poles, they begin to uncoil, the nuclear membranes begin to reform, and the cell is completing cytokinesis, the division of the cytoplasm. Now, I mentioned our cytoskeletal elements. There's more than just microtubules that are functioning here. And in cytokinesis, what is happening that's causing these cells to pinch apart, we're using the microfilament cytoskeletal element. In fact, actin. Now, I do this experiment far too often. And you don't have to raise your hand if you've done this experiment too. Every now and then, I'll kind of have to dress up. And every now and then, I'll have to wear a dress shirt that's tucked in. So I got to find a belt. And every now and then, I go to my closet and I put my belt on, and I swear my wife's putting one that's too small in there. Because I know I can't have eaten that much. Right? The belt is too tight. Well, I want you to envision what's happening to our daughter cells. They've got a belt of actin around them. And as that cell gets into late anaphase and moving into telophase, that belt of actin begins to squeeze. And it begins to squeeze. And that belt of actin gets smaller and smaller and smaller until, guess what? The cells are cut or sliced apart. And basically the phospholipid bilayers, they just fuse together, seal the wound, if you will, and you get two cells from the one. And that's the process of cytokinesis. You know, they used to do tonsillectomies kind of that way. Has anyone had their tonsils taken out? What they used to do, they used to get a wire lasso. It was a hot cautery wire. Because what separates your tonsil from the muscle of your throat is connective tissue. I mean, there are blood vessels and some nerves going there too, but you're asleep. Doesn't matter, right? But they will put this hot looped wire around the tonsil as close as they could to the throat, and then they would just slice. And the tonsil would just pop right out. And the hot wire would cauterize all the blood vessels as it was slicing through. Kind of the same thing happening with our actin. It's just a loop that slices completely through. And we get two daughter cells. And that's a process called cytokinesis. Makes you want to get your tonsils taken out, right? All right, that was mitosis. How are we with mitosis? Anything that we need to look at again? If we need to clear something up, let's do it before we go to the next one. Are we good? What are the 4 stages of mitosis? PMAT. Let's just stay simple. We can learn the big words by next week, right? PMAT. Prophase, metaphase, anaphase, telophase. What's the characteristic of metaphase? M means what? Middle. What's happening in anaphase? Moving apart. What's happening in telophase? We're finishing up. What's happening all before is just prophase. See, once again, this stuff seems and sounds scary, but it doesn't have to be. But you've got to understand those steps because meiosis is 2 divisions, one after another. Very similar to mitosis, but different enough to make it fun on a test. All right? So, meiosis is called a double division. Pardon me. I like to think of it, and write this down somewhere. I like to think of meiosis as a reduction division. In mitosis, we started with a parent cell that had 46 chromosomes. How many chromosomes does each daughter cell have after mitosis? 46, correct? Cloning. In meiosis, when you finish meiosis, you start with a parent cell that has 46 chromosomes. How many chromosomes does each one of the resulting daughter cells at the end of meiosis have? 23. You cut the number in half. That's the reduction part. And the way you reduce is you do basically a nuclear division twice. Are we good? Okay. Now, that's only going to happen in our cells that are going to form the sperm and the egg, and those are collectively called cells that function in gamete formation. Human gametes are sperm and egg. Big fancy name for sperm and the egg. So in this case, all of our somatic cells, those body cells, they have 2 copies of each chromosome, right? Those homologous chromosome pair. And we say that's 2N. What was that? What ploid was that called? Diploid. So at the end of meiosis, since we've only got 23 chromosomes, we're only going to have 1 copy of each chromosome. That's 1N, or haploid. Now think about this. You're going to divide 23 of your chromosomes are going to go into an egg or into a sperm. Are all 23 of the chromosomes you're going to put into that sperm, are those all the chromosomes you got from dad? Or are they all the ones you got from mom? It's some shuffled up combination. So do you see why you don't look like either your mom or your dad? And you don't look like your grandparents? Because every generation that genetic material gets shuffled up. You are a blend of mom and dad. And to what degree? It's absolutely random. It's just a lottery. I mean, you could in fact get those 23. Those could be the 23, that you got from your dad that you're giving to your child. But the other partner, those 23 are coming from her mom, her dad, or some combination of that. So that's what keeps the variability of the species. That's what helps prepare us for some horrific disease. Make sure survival of the species, we don't get all wiped out. If we started cloning ourselves, now think about this. The people that would go select the sex of their baby, right? Okay. We're getting to the point that we can pick and select physical traits. Okay? If we got to the point that you could pick out all the traits of your child, you just go to the baby store, sit down at a kiosk, start typing in all of your stuff, say, okay, that's the baby I want, right there. What's everybody going to want? The perfect baby, right? Who's going to go in and go up to the counter and go, what you got on sale? You got anything on clearance? Nobody's going to do that. So if we all want the perfect baby that's got the perfect genes, that's got the highest IQ, no mutations, no resistance to disease, what happens if a new disease comes out that can kill all those perfect babies? We're screwed. So that variability that happens naturally and randomly, that ensures not survival of the individual necessarily, but survival of the species. Farmers learned that a long time ago when they wanted the best of the best crops, and a blight comes along and wipes everything out. That's why all farmers don't always grow the same stuff next to their neighbors, and they don't always grow the same stuff year after year. There's our homologous chromosomes again. We got that, right? The 2 copies of the same chromosome, the same genes. We call them homologous chromosomes. So 2 divisions of meiosis. They very technically term these meiosis 1 and meiosis 2. Guess which one is first? Meiosis 1. Now, I want you to understand the difference between meiosis 1 and meiosis 2. In meiosis 1, and it's very hard to word this because I don't like using separate there. So I'm going to paraphrase this a different way. And you might want to change it in your notes. In meiosis 1, homologous chromosomes are segregated into 2 different daughter cells. Homologous chromosomes are segregated into 2 different daughter cells. So, how many sister chromatid pairs do we have in our beginning cell? How many sister chromatids do we have in our beginning parent cell? 46. All right? Now remember, those are sister chromatids But we've got those sister chromatids that are both from number 1, number 2, number 3. So what's happening in meiosis 1, 23 of those sister chromatids are being moved to one side of the cell. The other 23 sister chromatids are being moved to the opposite side. We haven't pulled anything apart. That's why I don't like that separate. We're not pulling any sister chromatids apart. We're just making sure that this daughter cell is going to be able to undergo what is basically just like mitosis there. And when those get separated, those two daughter cells are going to be exactly the same. These two daughter cells are going to be exactly the same, but they might not be the same as each other. Okay? So, meiosis 1, homologous chromosomes are segregated, moved apart. Now that's not the end of the division. It's a reduction division. It takes 2. And so in meiosis 2, each sister chromatid pair are aligned along the middle and pulled apart. So it's only when we get into meiosis 2, and each one of these are going to have PMAT. You're going to have meiosis 1, PMAT 1, meiosis 2, PMAT 2. And only when we get to anaphase 2 are we going to produce individual chromosomes again. We're working with sister chromatids all the way through the first meiosis and halfway through the second meiosis until we get to that anaphase 2. Then we pull them apart. Okay? I think we got pictures as we walk through this. Oh, yeah, here's a big deal. Typically, when we go through mitosis, interphase, mitosis, we have that S phase where we get replicated DNA. We're not going to replicate our DNA here. We're going to go through the 2 divisions with no DNA replication in the middle. That's what lets us cut it in half too. So that's an important step. All right? So let's do meiosis 1. Again, see, prophase 1, metaphase 1, anaphase 1, telophase 1. If you've got PMAT down, you're good with meiosis and mitosis. And you see it's a lot of the same stuff. In prophase, we're going to get rid of the nuclear membrane. Our sister chromatids are going to condense. The spindle fibers are going to form. In metaphase 1, we're aligning our sister chromatids along the equator, but notice what's happening. We don't have 46 lined up in a row. We've got 23 pairs side by side along the equator. Because one of these homologous chromosomes that are in fact sister chromatids, see, it gets confusing. It's going to go in this direction and the other one's going to go that way. But while they're along the middle, metaphase, anaphase 1, the homologous chromosomes are segregated. Again, you see we've not pulled these apart. They're still sister chromatids. And we get to telophase, cytokinesis is going to occur. We're not really going to form a nuclear membrane because we've got to go through another division right after that. But you see we still have sister chromatids here. We just have half as many sister chromatids as we started with up here. Does that make sense? So again, looks a little bit like what we saw in mitosis, but it's not the same. Now, here's one thing that I want to touch on that happens in this prophase 1 that doesn't happen in mitosis and it doesn't happen in any other phases. It's a process called crossing over. And it's where you actually get exchange of DNA between homologous chromosomes. This is where a chromosome you got from dad, it's going to exchange some of its genetic material with a chromosome you got from mom. And so the result is going to be a chromosome that's never existed before with a unique combination. So here's how the process sort of works. Here are our homologous chromosomes, and they're duplicated sister chromatids. Well, 2 of the arms get tangled up. We're going to say the purple is mom and the blue is dad. So 2 of our chromatids get tangled up, and what happens is there is a break in the DNA, and when the enzymes come in to fix the break, do you see what's happened at the bottom? Here, we've got this segment of dad's chromosome stuck on mom's bigger chromosome. And that segment of mom's chromosomes now here on dad's bigger chromosome. That is, again, another shuffling of genetic material, creating, in this case, two chromosomes that are going to end up in daughter cells that have never existed before in the history of mankind, that could potentially go into an egg or a sperm to form your offspring. So again, a nice blend of the genes between mom and dad. Now, this point at which the DNA gets tangled up, it's called crossing over. This has always been called the chiasma or the chiasmata. I guess to get politically correct, I always kind of laugh at this. To get politically correct, they have renamed this the holiday junction. We can't say Christmas anymore. We have to say holiday. Well, we can't say chiasma for cross anymore. We say holiday junction. I say chiasma because it's cross. It's what it looks like. Don't make it more complicated. So not only when you're forming the sperm and the egg are you shuffling mom and dad's chromosomes that you got to put into your sperm or egg, crossing over also occurs to add a level of randomness and variation to help, again, the species survive. So here we have our sister chromatids. These are our homologous sets. Maybe I'll just call them homologous sets. Maybe that's a little bit less confusing. Notice here we have a homologous set and we've had crossing over occur. So we've mixed up the genetic material, but they're all along the midline. So what do we call it? Metaphase. Metaphase 1, homologous chromosomes align at the equator. 23 on each side. They're not lined up single file now. They're lined up in pairs. You know, I asked you this already. Will all dads go one way and all moms go another way? No, completely random. And this just shows you some of the possibilities that will happen. All right. Anaphase, let's finish meiosis 1 and then we'll be done. Anaphase, we're moving those sister chromatids in opposite directions. We have segregated them to the poles. Again, like we saw in mitosis, spindle apparatus is elongating the cell, pushing it further and further apart because we're going to undergo cytokinesis. It's going to be happening right here toward the end of telophase. Now notice in telophase 1, each daughter cell has 23 duplicated chromosomes. There are 23 sister chromatids in each one of these daughter cells. Now, this gets tricky because scientists say even though they're sister chromatids, so there's two attached to each other, and so in fact you've got 46 chromatids present. There are only 23 different ones, so at this point we say that our daughter cells are in fact haploid. Even though they're still attached at the centromere, still in fact sister chromatids. We call that meiosis.